Hello, myself Dr. Prabhakar Bhanandas Ghorpade, Assistant Professor, Department of Veterinary Biochemistry, COVAS Parvani. So today I am going to talk about my department, Department of Veterinary Biochemistry. In Department of Veterinary Biochemistry, we have postgraduate research laboratory. So in the postgraduate research laboratory, we teach how research is being done for the PG and PhD student, as well as this laboratory can be used for the UG student, undergraduate student. When we talk about research to the UG as well as PG student, we talk about central dogma of molecular biology. Under central dogma of molecular biology, we have steps like DNA is getting converted into the DNA, that is DNA replication, DNA is getting converted into the RNA, that is transcription, RNA can use the protein, that is a translation process. We try to explore this molecular biology technique in our biochemistry lab. So when DNA can be getting converted into the DNA, that is the replication process. We can show to the student the replication process with the help of polymerase chain reaction. So we have equipment required for this technique, that is replication. We have thermocycler and we have centrifuge machines. The related technique which are required for the showing the replication is present in our department. As well as when we talk about transcription, DNA getting converted into the RNA. So this transcription process we can do in our laboratory, that is gene expression techniques. For that we have real-time PCR machine. So with the help of reverse transcriptase enzyme, the consumable chemicals required, we can show how gene expression can be done in the particular cells or the tissues. Then we have RNA to protein, that is protein expression. So protein expression we can do in our laboratory. So that is the flow of the equipments we have maintained. So basically we can do most of molecular biology lab work in our laboratory. We have UV transilluminator, then we have for detection Western blot equipment. So if you have some clinical sample and you want to detect for example any virus sample, rabies virus you want to detect from dog saliva, whether it is present or not. So we can use this PCR technique to identify whether that virus is present or not. Or otherwise, if you have particular antigen present in the infectious disease, you can detect this with the help of the antibody testing, that is Western blot. With the help of Western blot, we can detect that antigen. The clinical sample we can analyze with this one. So these are the different flow of equipment. So when we are going to see our laboratory, we have different equipment. We have ice flaking machine, which will be required for transformation or transfection to maintain the cool temperature of the bacterial cell or the animal cells, or any biochemical reaction which require the controlled temperature, that can be done with the help of this ice flaking machine. Then we have high precision electronic weighing balance, which helps to measure the precise chemical weight. Whenever we measure the chemical weights, so during that process we have try to keep the related chemistry chart in our laboratory, like chemical structures or periodic table. So whenever UG student comes to me and he asks me how to calculate the molecular weight, I show him the molecular weight periodic table, and then from periodic table they get understanding how 1 molar NaCl can be calculated. So based on the molecular weight of sodium and chlorine, we can add it into up to the 1 liter and we can make 1 molar. So likewise, so basically one technique we are doing, the related charts we try to keep it. When we say water, or when we say alcohol or glucose, we try to demonstrate the molecular structure of that chemical entities. So for that purpose we have molecular kit with us, means the molecular models we can generate, the carbon we can join, and we can make the glucose for them, or we can make the water molecule for them, or alcohol molecule for them. So this we can find in our laboratory how this can be done. When we say we have to incubate the bacteria or particular tissue sample, we have this water bath with us, or bacteriological incubator we have with us for handling the infectious agent, or to maintain the biosecurity protocols. We try to do everything under the laminar flow. So we have 2 laminar flow equipments in our laboratory. For culturing the bacteria we have refrigerated shaking incubator. For separating the individual biochemical components, we have this centrifugation machine, cooling centrifuge machine. So if you want to do the bacterial streaking for showing its biochemical characteristic of the microorganism, we have the loop and the stirrer, this equipment, and whenever any infectious agents or any biological entities handled, likewise serum or any bacterial sample, so in that case we have autoclave with us. So this with the help of that autoclave, we try to keep everything sterile. So before the experimentation and after the experimentation, all things should be kept in the sterile environment. So these things we try to maintain. That is the sterility of the process. So very important for the students as well as the instructor point of view, we try to maintain. When we see our biochemistry lab, we try to maintain the instructions from the students. So the visual instructions like how you should behave in the lab. So you should wear proper apron, we should wear the shoes, we should have the glasses on when we are handling the biochemistry equipments or the chemicals, because we are dealing with the dangerous chemicals sometimes, like concentrated H2SO4. So during this time, you should have the face shield, you should have proper gloves, and then you should handle this equipment. How you should handle these equipments? You should not keep the heating things on unattended. Any experiment, we should not eat anything in the laboratory. These instructions we try to convey to the students. So these are the important things in the biochemistry laboratory. And during the so this is about our PG laboratory. So basically under the PG laboratory, we try to provide the services to all college level students. So most of PG or PhD students comes to our laboratory. They comes from either the PCR or the serological analysis or any experimental techniques which involve the biochemistry parts. They comes to our laboratory. So we try to provide the services. In addition, so the next thing we have is in the TVCC, Teaching Veterinary Clinical Complex. In that one we have the clinical biochemistry laboratory. Under that clinical biochemistry laboratory, we have fully automated biochemistry analyzer. Next, the hematological analyzer, that is also fully automated. Next, blood gas analyzer, and then urine analyzer. So talking about the fully biochemistry analyzer, the purpose of the biochemistry analyzer is a serological testing. So we have total protein, albumin, globulins, cholesterol, AST, ALT. These estimations, these for UG student we do it by the handheld pipetting. But for maintaining the accuracy and the consistency in the result and to avoid the manual error which can be done when the person handles does pipetting. So in that case we have fully automated biochemistry analyzer. So around the 50 tests can be done in the 1 hour with that biochemistry analyzer. So that's this we have intentionally kept it at the clinical position, so that whenever clinical animals come in the clinic, the students, internship student or the postgraduate students of the clinical subjects which are available there, so they can do the analysis there with the help of the fully automated biochemistry analyzer. Next we have hematological analyzer. So for hematological analyzer, the student can take the sample from the clinic and directly they can place the sample with this anticoagulant added to that machine, fully automated machine, and it can use the whole sort of parameter like RBC, WBC, the differential leukocyte count, so platelet count. Likewise, parameter can be given with that equipment's fully automated biochemistry analyzer. Next we have is a blood gas analyzer. So animals comes with a different complaint like acid-base imbalance. So whenever there is a respiratory acidosis, respiratory alkalosis, or the metabolic acidosis, metabolic alkalosis, acidosis or alkalosis, in that condition it is very essential that you measure the amount of which gases are in excess or the lower. That is whether there is a higher partial oxygen pressure or higher partial CO2 pressure, or what is the pH, what is the electrolyte concentration in the blood. So instantly student can take the blood and directly they can feed the blood into the machine, blood biochemistry analyzer, blood gas analyzer, and they can analyze this with the blood gas analyzer, the amount of gases and the electrolyte and the pH there. Next, animals urine can be analyzed with the urine analyzer. So we have strip based urine analyzer present there. You put the strips in the urine and you feed that to equipment, and all your urine parameters can be detected with the urine analyzer. So basically the biochemistry, when we talk about our biochemistry component, we have one animal. The animal is eating something, some biochemical changes is happening in its serum, protein level and other things, cholesterol. It can be detected in the UG laboratory, undergraduate or our clinical biochemistry laboratory. So the animals blood we test animals serum, we test animals blood gases, then we test urine. So most of things we try to cover under the biochemistry, being the basic department. So our biochemistry department comes under the basic sciences. So basic sciences, we try to maintain the basic flow. We try to maintain the flow from the genetic level, DNA level, to the molecular means microscopic level to the microscopic level, molecular to the macroscopic level. At the molecular level we study the DNA, at the macroscopic part we study the proteins which are present in the serum or the different metabolites. In addition, department is currently trying to have the EHPLC equipment. This EHPLC equipment, with the help of that equipment, the metabolic analysis can be done. So this metabolome analysis can be done with the HPLC equipment. And for UG students, some very basic things we try to maintain at our place. Like many students, they will be requiring the distilled water in the research. So how to get the distilled water? So getting the distilled water with the conventional technique is very tedious process. Means we have to heat this double distillation things. So for basic experimentation purpose, we try to maintain this molecular biology grade water for them. So each day they can take 15 or 20 liter water from our department for their experimental purposes. So we try, in that way the department tries to serve the UG student, PG student, PhD student, as well as the farmers which comes to our clinic to help them in the processes, means learning process, either the learning process or the animal care process. So in addition, these are the different. So basically we have maintained a lot of equipment repository with us, with the knowledge repository in addition to that. So when we maintain this repository, to our point of view, we try to display, we don't try to keep that equipments and knowledge with us. Equipment point of view, we try to give the equipment services to the students as well as the farmers. And knowledge point of view, we try to organize the trainings. For our undergraduate students, we have organized the DBT Star College schemes. So DBT Star College scheme. So under this one, we show the equipments, how equipment working can be done, how these things can be done, means experimental processes can be done. As well as we try to train the faculty, all India level faculty, with the help of the biochemistry training. Already we have conducted 2 10 days biochemistry training. Though basically for this training we try to get the faculty from the other places. So the most of all India level faculty, they were involved as a guest faculty in our training process, and they have very nicely delivered the lecture. So I am thankful to those elite faculties for the contribution they have made for our training programs. Now talking about the services. Now when we say we have this DNA to RNA, RNA to protein, and those are lot of equipments I talked about. So how those equipments came to our department. So basically we have in our department we have 2 schemes. DBT Star College scheme. In that scheme, the DBT's motto is that they want students to get exposure to the molecular techniques as well as the equipments which are not available in the college level. So in that way, in the DBT around 50 equipments we have purchased, small equipments we try to purchase for the student. So these equipments were given by the DBT under the DBT Star College fund. So around 10 lakhs funds we got in the DBT Star College scheme for the equipments. Then 3 lakhs for the 2 times we got for the consumables. So in that way we have got a lot of support from the Department of Biotechnology, Government of India. So I am thankful to DBT also for giving us opportunity to serve the student in that way. Next, we have other research scheme, Science Education and Research Board, ESRB New Delhi. There, under this one, I am running one research project. In that research project, we try to generate some mutants of Salmonella and evaluate those. Under that scheme also, we have taken around 20 lakhs worth equipment like refrigerator shaking incubator, then for DNA testing this nanotop equipment. So this we try to get this from the ESRB. So this they have given us opportunity to run this research scheme, and this scheme is basically a research scheme. The research output will be there, but basically this scheme is serving very nicely to the PG and the PhD students. So this most of PG and PhD students have benefited from our equipments from ESRB. As well as UG students, they have got benefit from the ESRB's research scheme equipments, as well as knowledge center which we can gain. So I am very thankful to the ESRB New Delhi for their research scheme. Next, ICAR. So ICAR routinely as a university, MAPSU University and COAS Parbhani College, we got a lot of support from the Indian Council of Agriculture Research, New Delhi. So from their contribution, we could purchase the different consumables, chemicals, and glassware in our laboratories. So I am very thankful to the ICAR for their contribution in our our college and for the veterinary sciences community. Next, our beloved associate dean and the HOD of the department, they have helped us to get the grant from the revenue receipt of the college whenever required. So they have very kindly given the grant for the consumables, chemicals, and the glassware whenever we try to get the grant as per necessity in our department. So I am very thankful to them also. So these are the different schemes and the different sources of the things we have in our biochemistry department. Under Department of Biochemistry, we try to have some bioinformatics setup also. So whenever students comes to us, when we teach to student the nucleotides, in that case we teach them nucleotides. So nucleotides, DNA sequence, RNA sequence. So students basically they can actually do the research work like PCR. In that case, they can actually do the research, as well as this nucleotide sequence they can visualize when they visit the National Center for Biotechnology Information (NCBI) website. So for NCBI website searching, we try to give the student the knowledge regarding the bioinformatics. So what knowledge regarding bioinformatics we can give to the student? We can teach them sequence analysis, how to search the sequences, how to align the sequences, how to get the protein three-dimensional structure, how to do the mass spectrometry analysis, ENMR analysis. Likewise, different bioinformatics setups and analysis we try to impart to the students. In addition, department already has purchased under the DBT Star College IoT setup, Internet of the Things. So we try to maintain the Internet of Things setups also for like color-based test detection or any the advanced techniques. So the Internet of Things is very recent thing. We try to give them a little bit of programming knowledge or how to program the things and how to make the automated equipments. In addition, for the statistical part, we try to teach them R or the Python or statistical analysis with the help of R whenever any biochemical test is there for PG student. So basically, I also try to learn this R, Python, and this artificial intelligence technologies. So as to the purpose is to teach the student. So the student can do this automated analysis with the programming and all those things. So basically, the students who have interest in these techniques, they can take knowledge from us and they can have very good research articles and the papers with the help of techniques which will be available with this R and programming languages and the statistical analysis. In addition, we have PG and PhD facility in our department. So the PG students already they have done research on A1, A2 milks, the beta casein. that was molecular biology related research, and now the PhD student, we have one PhD student also. So they are also actively contributing to the research and the development in the department of the veterinary biochemistry. So in the department of, in addition, when we teach the student, so we try to incorporate with different teaching aids. Like when we see, you will see the UG level of UG laboratory of veterinary biochemistry. There we try to hang some charts there for student. Whenever some class is going on, they can see the surrounding around them. So it creates a very knowledgeable environment. If they are even visiting department, somebody is visiting department, they can see the charts, they can try to understand something. In that way, we try to maintain the UG level display and the charts for them. When we next for NPG and PhD student. So basically, when we look at the veterinary biochemistry department, basically our department is a basic science department. Like in the medical sciences, in the veterinary sciences, this the same things we teach there as a biochemistry. So it is basically connecting link with the paraclinical department, basic science department, and the clinical science department. So initially the learning course can be bit difficult from the student point of view, because we have a lot of structures and biochemistry reactions, but we try to teach them in the understandable way with the help of PowerPoint presentation, molecular models. And the understanding of these biochemistry models and biochemistry reactions is very important for the student point of view, because when we are as a student, you are going to go in the department of pharmacology. In that case, you will have some drug which will be acting at the particular pathway, and it will be blocking that particular pathway. So pain will be reduced. For example, animal is having pain, so you are blocking some cyclooxygenase pathway, and the pain of the animal is decreased because of that pathway. So in that case, the basic understanding is that you should know the biochemistry reaction. Then you can know where it blocks, particular drug blocks, or the inhibitor blocks, or enzyme inhibitor blocks. So these things is very important. So our course is basically a founding stone in the veterinary biochemistry. So based on, if you have this stone proper, then you can have the pharmacology knowledge proper. And if you have pharmacology knowledge proper, then you can have the medicine, surgery, other things knowledge proper. So basically the knowledge is step by step. So we try to maintain the first step in the veterinary biochemistry in our department of veterinary biochemistry. So thank you everyone for your nice and persistent listening to this video. Thank you very much.